Okay, this is part 2 of activation of acid production. What I'd like to discuss now is a principle of what's called potentiation. Now, potentiation is when two effector molecules are going to act in what is considered a synergistic manner. So one will create an effect, and another one that creates the same effect will be essentially additive or more than additive. Now, what does that mean? We saw in part 1 that gastrin and histamine are two effectors, activators of gastric acid secretion. So if we look at the rate of gastric secretion, and compare this rate with increasing concentrations of gastrin, the hormone coming from the G cells that induces the acid production, acid secretion, we will see that gastrin has a very small effect at low concentrations, and then as the concentration rises, now we get the full effect of gastrin. So there is a certain threshold concentration that is required in order to get gastric secretion activated. Now, if we didn't use gastrin and we used the hormone, the effector histamine, we would see the same type of effect on the rate of gastric secretion. So if we have low concentrations of histamine, very little effect, and then as we increase the concentration of histamine, we now see that there's a positive effect on the activation of acid secretion. Now, the concentrations that are working at the thresholds between gastrin and histamine are not equal. Gastrin is a much stronger inducer of this, but this is just mostly descriptive. So now, what happens if we add gastrin and histamine together? We are going to now get the effect what is called potentiation. And potentiation means that there is a much greater effect when both hormones, both effectors, are added together. Both effectors are creating the same situation. They are increasing, they're activating acid secretion. But when you add them together, you get a shift in the curve to the left, which means lower concentrations have the same effect as higher concentrations. That's called potentiation. Now, this is a good time to start introducing pharmacology, and let's look at a familiar over-the-counter medication that is an antacid. This inhibits acid production, cimetidine, and it does so because it is an H2 receptor antagonist. So this binds, cimetidine binds to the H2 receptor and prevents the binding of histamine to that receptor and thus blocks the effect of histamine on acid secretion. So if we give a patient cimetidine, what we are going to see is the rate of gastric secretion is going to be blunted, and it's going to be blunted back to the level of gastrin because the patient is still producing gastrin. This is why cimetidine tends to be considered a weak inhibitor of acid production because it only blocks one of the effector, and in fact it blocks the weaker of the effector. But what it's blocking is the effect of potentiation. So we still get some acid secretion, which we'll see in a bit. It is still very important for a function of the stomach's activity. But we do have a reduction in acid secretion, which as we know, can help alleviate the symptoms of acid reflux. Now, the other drug we'll look at later is omeprazole. This will be an absolute block on acid secretion through the proton potassium pump. But before we look at that, I want to get into the just some of the second messengers that are involved in the control of acid secretion. So if we look at our parietal cell here, that is going to be inducing the acid secretion. Here's our lumen of our stomach. Remember, we have essentially 3 different activation signals. The first one we have here is through acetylcholine, through the vagal stimulation. And remember, the vagal stimulation begins in the cephalic phase of digestion. Acetylcholine will, of course, trigger the increase in calcium. And the calcium will be the effector, second messenger, that will control the rate of acid production. We'll see that in a second. The other receptor we have is CCK isoform B. This is the receptor for gastrin. This receptor also works through the similar signaling properties to increase intracellular calcium levels. And the third effector we have is, of course, the H2 receptor. And this is the effector, this is the receptor for histamine. Histamine will work through adenylyl cyclase, which of course will increase the cytosolic concentration of cyclic AMP. Now, what do these second messengers do? Recall in the last part 1 I mentioned about parietal cells have these intracellular vesicles located, clustered mostly, concentrated mostly near the lumen of the plasma membrane. The calcium will stimulate the fusion of these vesicles with the plasma membrane. When that happens, the potassium proton ATPase transporter is now exposed to the luminal surface. Before, it was sequestered in one of these vesicles. Upon fusion with the cell surface, it is now placed in the location where it can pump protons out into the lumen of the stomach and decrease the pH. This is a good point to mention the next drug, omeprazole. Omeprazole is a proton potassium ATPase inhibitor. Now, many of you probably know that omeprazole is often, most of the time now, the drug of choice, the pharmacology of choice for inhibiting acid production. The reason for that is that omeprazole is an irreversible inhibitor. Now, what does that mean? Now we have to go back to a little bit of our kinetics and our biochemistry. The ATPase antiporter, once it's exposed on the cell surface, is now in a position to be inhibited by omeprazole. This drug binds covalently to the transporter. Binding covalently means that it is non-dissociable. So how do we remove the omeprazole from the transporter when we want to dilute the effects of the drug? Because it's non-dissociable, covalent interaction, this whole protein complex has to be degraded in the proteasome. You actually have to degrade the entire pump and get rid of it, and then restoration of the transporter is through what's called de novo synthesis. De novo synthesis is essentially gene transcription, translation into protein, and restore the protein through biosynthetic mechanisms. Now, Dr. Shuttleworth will be going over the exact mechanism of omeprazole, which is very interesting, that omeprazole becomes activated by low pH. And the drug is inactive at neutral pH, and it becomes active when it gets into a low pH environment. What that means is that the patient takes the omeprazole orally. It's absorbed into the blood, which of course is pH 7.35. It is transported into the parietal cell and becomes localized to the vesicles, bound to the transporter. And this is the proton potassium transporter. Once the vesicles fuse with the plasma membrane at the luminal surface, they are now exposed to the low pH because the low pH has already begun in there, and the drug changes its chemical nature to form a covalent link with the proton potassium transporter. Once this happens, the pH activates it, it binds covalently, the transporter is now shut down.